Okay. Right. What I said last, the hydrogen ion can change the pH because too much hydrogen ion can decrease the pH. It become more acidic. That is not good because enzyme can't work in acidic conditions. So then only way to escape this hydrogen is through this ATP synthase complex. Because ATP synthase complex can transfer hydrogen ion out. Only ATP synthase can do this one. So it can transfer hydrogen out of the thylakoid, right? So when it remove out of the thylakoid, now the energy is again changed. So energy is again transfer. This time, this energy transfer to make ADP plus phosphate to ATP molecule. So ATP molecule also formed. How it formed? Hydrogen ion move out of the thylakoid into the stroma, and the energy trapped to create this gradient again transfer to the ATP molecule. So now what are the products? Number one, NADPH. Number two, ATP. In addition to that, oxygen is also formed. Okay, this is the end of the light-dependent reaction, linear electron flow. In that case, you have noticed the electrons, these electrons moving to PS1 complex. So it is a linear. And these electrons from PS1 move into NADPH. So it is a straightforward linear reaction. There is no recycling or cyclic process happen. In the first part, PS2 complex, hydrogen ion pumps in and photolysis take place. And PS second part, PS1, only the electrons move into the NADPH. It forms NADPH. What are the products of light-dependent reaction? NADPH, which is energy-rich molecule. And we have learned previously ATP, energy molecule, energy currency, and oxygen as a byproduct. Now I took this question again from the past paper. Which should the following biochemical process require ATP? Photolysis of water during photosynthesis. This word not in the resource book. So only few things we have to learn in addition to the resource book, but most of the things are coming from the resource book. This is one example in the exam and where we cannot find photolysis word. Okay, next, if you have any question, you can ask about this process, linear electron flow, and we have then cyclic electron flow. The cyclic electron flow, its significance is very less, and most of the time what we can observe in the plants, this linear electron flow, because it produces oxygen, it produces NADPH, and it produces ATP. The cyclic electron flow is a term we only used to understand there is another one existing like this, but its significance is less. Why it is significant is less? If we look everything what we learned so far, this is the thylakoid membrane, photosystem 2, photosystem 1. Light is absorbed by the PS2, and that electron is moving to the electron carrier complex, and the electron moving to PS1. In PS1, light is absorbed, this electron is moving again to the complexes and NADPH formed. But cyclic electron flow, only PS1 is active now. Light is absorbed by the photosystems. The chlorophyll excited its electrons. So electrons with the energy released from the photosystem. Now this electron has energy, right? Because the energy from sunlight transferred to this electron. This time what happened to the electron? It is now moving to the electron carrier proteins here, and then it is moving to the other side of electron carrier proteins. Now when it comes this pathway, so you can see how it works. It just enters one electron carrier, and after that it goes to the PS2 electron carriers. So then it moving down, and when it come to here, energy lost. Because I highlighted this one to say it has energy, but here no energy. So then it come back again to the complex to stabilize the complex, because this complex lost electron. Now electron moving back to stabilize the complex. This again pump hydrogen, and establish hydrogen ion gradient. So this hydrogen ion can now escape through this ATP synthase complex, and in this case again it can make ATP. Only one product, it's only the ATP produced. Cyclic electron flow produce only ATP. There's no oxygen, no water splitting, no photolysis, no NADPH. So it can produce only ATP, and you can see here the cyclic process. This electrons flow is cyclic. That's why we call it cyclic electron flow. Most of the time this is not a target for the exam. This is just as traditionally in any textbook we have to learn a little bit of a cyclic electron flow and not used to make questions because its significance is very less in photosynthesis. It doesn't produce oxygen and it doesn't produce sugars, carbohydrate, lipid, like it is not fit into the original definition. It's a metabolic process in which light energy is converted into trapped and converted into chemical energy such as carbohydrate, lipid, and proteins. So here we can't see that kind of a thing. So its significance is less. Maybe a certain situation, plant produce this cyclic electron flow, and maybe a prokaryotic like organism just to get their ATP, they can use this pathway. Other than that, this significance is very less. Okay, we have completed the light dependent reaction. If you have question, you can ask. If not, we have to move into the light independent reaction. Okay, light independent reaction. Now, light independent reaction is also known, also called Calvin cycle. This is also called Calvin cycle, and Calvin cycle take place in the stroma. So far, everything happened in the thylakoid membrane. So this is the thylakoid, and this place is the stroma. Okay, now the light independent reaction or Calvin cycle start with first reaction. First reaction is uh, a compound called ribulose bisphosphate. Ribulose bisphosphate. We try to write the abbreviation for this one, RUBP. RUBP is the abbreviation for ribulose bisphosphate. And ribulose bisphosphate has 5 carbons. It is a compound with 5 carbon number. So ribulose bisphosphate going to react with carbon dioxide. Carbon dioxide has 1 carbon. And this reaction is catalyzed by enzyme Rubisco. Usually, this is the abbreviated form of the enzyme. This enzyme complete name is ribulose bisphosphate carboxygenase oxygenase. Ribulose bisphosphate carboxygenase oxygenase. But you can write as Rubisco. This is a standard form, and no harm to write this abbreviation. It is okay to write the abbreviation Rubisco as the enzyme name. Now this 5 carbon and 1 carbon joined together makes 6 carbon compound, and this 6 carbon compound has no name. We just write it's a unstable intermediate product. Unstable intermediate product. And this unstable intermediate product split again to 3 carbon molecule, This three carbon molecule is called 3 phosphoglycerate. 3 phosphoglycerate abbreviated by TPGA. 3 phosphoglycerate. Okay. So let's see again this reaction. RUBP, carbon dioxide, make unstable product, and then it split into 3 carbon, 3 phosphoglycerate. This is catalyzed by enzyme Rubisco. So this first reaction is called carbon dioxide fixation. Carbon dioxide plus ribulose gives 3-PGA. Enzyme is Rubisco. This reaction is called carbon dioxide fixation. After that, 3-phosphoglycerate reduced to glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Now this reaction is again important because this reaction need energy, and energy is produced by the ATP and NADPH. So these two products are coming from light dependent reaction. So ATP and NADPH provide from the light dependent reaction. Now glyceraldehyde 3-phosphate select two pathways. One is the regeneration of ribulose bisphosphate. This required again ATP from the light dependent reaction. And the second pathway is glyceraldehyde 3-phosphate can move into produce glucose. But we are not learning that pathway in details. We just say glyceraldehyde 3-phosphate can produce glucose. So this is divided into fixation, reduction, and regeneration. Fixation, reduction, and regeneration. So we are not going to learn again how many carbon dioxide molecule required and how many glucose produced with carbon dioxide. Like details are not required. Only important things we have to remember: the products, ribulose bisphosphate, the enzyme, and 3-phosphoglycerate converted into glyceraldehyde 3-phosphate, and recycle this one back to the ribulose bisphosphate. And ATP and NADPH required from the light dependent reaction. This is very straightforward. We can remember the three reactions in fixation, reduction, and regeneration, and where the energy is important. NADPH can provide energy and back to NADP plus. ATP provide energy back to ADP. Then the energy is used to make the cycle going on, and part of the glyceraldehyde 3-phosphate make glucose. And also in the syllabus, we don't learn how proteins form, how lipid forms, because if the glucose is starting point, this should be the main product for all the other molecules like lipid proteins, like DNA, all the molecules. Those pathways we are not learning. We just stop the Calvin cycle saying it produces glucose from glyceraldehyde 3 phosphate. Rest of the pathways are not important and we are not learning. So what are the product of a Calvin cycle? Basically the glucose. So energy is provided by the light dependent reaction. Now you can see, so far we have learned photosynthesis as a one reaction. That is carbon dioxide, water, it gives glucose and oxygen. Now we have seen this one reaction is not the simple reaction. It is resulted of many reaction like this. Right. Again, when you have to study, as you can see, this kind of a thing can be given in MCQs. Find the incorrect answer or correct answer. Then again, PS2 excited electrons. This electrons is moving like this. The sentences are given. Minor mis. They can change small thing and can be asked the questions. So it is better that you remember the whole process of light dependent reaction and also light independent reaction. Okay, if you check the notes, what is the function of mitochondria? What is the function of peroxisome? There is a one word written photorespiration. You don't know photorespiration, and we haven't learned photorespiration there, but we just use the word photorespiration. So, one of the function of mitochondria photorespiration, and also peroxisome photorespiration. Now, what is photorespiration and why it is important to understand? Okay, to understand this process, we have to little bit look what is the structure of the leaf, right? So, if you look the leaf, the top layer is the epidermis, epidermal cells. Then below that, there's another cell layer. These cells are called palisade mesophyll cells. Palisade mesophyll cells, and below that, then again the spongy cells, spongy mesophils. The spongy mesophyll cells ended up in another layer called lower epidermis, and lower epidermis there's two guard cell, and this one is opening is called stomata. And if we look where is the location of chloroplast, we can find a lot of chloroplast in the palisade mesophyll cells, and sometimes a little bit in the spongy, and we can also find in the guard cells. That means photosynthesis take place heavily in the palisade mesophyll cells and little bit spongy mesophyll cell and also guard cells. Okay, now where we get this water and carbon dioxide? So water can directly get from the soil and water transport to the cells. Carbon dioxide? Carbon dioxide is coming from outside. Okay. Where is oxygen is leaving? Oxygen also leaving from this place. Okay. Can you name another molecule which uses the stomata to move through it? Another molecule which can also move like this way through the stomata. Oxygen is going out of the cell, carbon dioxide is coming into the cell. Another molecule. Yes, Apurva is correct. It's water molecules. When the body is absorbed by the root, what is going up, then after that, this word is escape. So it is very important that plant need to absorb water because with the water, it can also absorb minerals and the minerals can transport up like nitrate like minerals for the metabolic reaction and excess amount of water is moving out as water vapor. So we can't see that it is water is going like a tap, but the water vapor form, the water vapor is going out. So it is important as a transport medium. It is also important to maintain the temperature. It can cool down because when the water going up, it take heat and then evaporate, and then the heat is moving out. This is called transpiration. The process is called transpiration. Transpiration is water vapor is moving from the through the stomata. It's the transpiration. Okay, let's see a condition. That condition is very dry and hot. If the environment condition is dry and hot, less amount of water, the plant can absorb less amount of water because water level is less in the soil. And if this is happening, plant loss water. In dry and hot condition, if transpiration take place in a maximum speed, plant loss water, plant can even die. It can even die because of loss of water. So only thing is it stop evaporation of water. So it can conserve water. So if plant wants to conserve the water, the stomata is like this shape. So we have to learn everything in the plant section. These are the guard cells, and guard cell can closed. This opening is the stomata, and the guard cell is closed. When the guard cell is closed, stop evaporation of water. But there is a problem of photosynthesis, the carbon dioxide. If the guard cell is closed, the stomata is closed, carbon dioxide level inside decrease. Carbon dioxide need from the air to start the Calvin cycle or light independent reaction. Then carbon dioxide when enters stopped. So carbon dioxide cannot enter. So photosynthesis can take place still with the available carbon dioxide. So whatever carbon dioxide entered, they can use to make the photosynthesis, but largely new molecules are not coming. And the process is not 100% black and white. That means when it's really dry, 100% closed throughout the day, nothing is happening. So it regulates, like it can little open, little close. So carbon dioxide level of carbon dioxide entry is reducing. If we take throughout the day, it is not the story that 8 o'clock stomata is completely closed in the morning, and then the stomata is open in maybe 8 o'clock in the night. No, this is not the case. It's like a very changing situation. It can open a little bit, it can close again, little bit open, like that. It's very fluctuating. So it's not regularly happening. It's not a very complete process. It disturbs everything. So overall, carbon dioxide level inside is decreasing, but photosynthesis also take place. So which create a situation oxygen level increase than the carbon dioxide level, because when it is closed, oxygen can't go out, oxygen trapped. Photosynthesis took place, remaining carbon dioxide, but it's closed that time and oxygen cannot now move. So net effect is level of oxygen little bit higher than the carbon dioxide. Oxygen level increase. Okay, now in enzyme section you have learned enzymes are specific to the substrate. Enzymes are specific to the substrate. In this case, what is the substrate? Carbon dioxide and ribulose bisphosphate RUBP. RUBP and carbon dioxide are the substrate. Enzyme is Rubisco. Rubisco is specific to the carbon dioxide and ribulose bisphosphate. But that concept is not always correct. When we say enzymes are very specific, in the previous lesson, we have to accept here that is not the always correct. That's why sometimes we use the mostly, generally, the most of the proteins are enzymes like this. Most of the enzymes are proteins like this. We use word mostly general like this. So in this case, we can expect a reaction RUBP, oxygen. The reaction of RUBP with oxygen catalyzed by Rubisco. When more oxygen is available, Rubisco can also catalyze a reaction between RUBP plus oxygen. Okay, we'll see what will happen after that.